Okay, guys, welcome to the first digestion video. So we just finished up with the kidney. We took 4 lectures to go over everything you need to know there. And we did do it pretty darn in depth, right? So digestion, I just found out from Dr. B is actually going to cover 2 lectures instead of just one. And I was a little worried because there's quite a bit in digestion. So today we're focusing on just the first half of the digestive system. And next time we'll talk about the second half. And I feel like the second half is a little bit more involved with the absorption of all these molecules we have to talk about. So it's really great for us to get to start here, something a little bit easier. I think this lecture will be kind of short, hopefully no longer than 5 to 10 minutes. Just kidding, that's not happening. But okay, so let's get started. So this is a great picture and we'll bring it back up later on. But this is just digestion in general, right? So we didn't want you to think of, well, we're going to teach you the digestive tract. All you need to know is how food gets into the body. We also need to know, like, how is it broken down, which is pretty much digestion, how it's absorbed into the bloodstream, how it moves, right? Like what the motility is, and then what are all the secretions. And I think the secretions are some of like the most easily testable material from these lectures, and also some of the best stuff for you guys to know just in general to understand how your body's functioning. So today we're talking a lot about the digestion. We'll have a little bit more of it next time. I'm not really going to cover any absorption today. That's going to be next time. And I really think that's the harder stuff. And motility we'll also get into, and a little, about half secretion. There's so many secretions, we couldn't cover them all in one lecture. So these are all the objectives that we're going to go over. So we're in the anatomy of the gastric system. So this will just be great for those of you who have not taken anatomy, just to understand what we're talking about. And I'll just do that very briefly. You guys need to know all the secreted molecules. So there's so many, and they're not just all proteins. Some of them are just acid, like hydrochloric acid. There's other enzymes that you need to know that help with digestion, that like if these things didn't exist, you wouldn't be able to digest any sort of food. So that's a big portion of these couple lectures. Like if I were you, every single time you hear of like something new, like cholecystokinin or gastrin or leptin or pepsinogen or aminopeptidases, any of those things, I'd write it down, get the definition, get where it comes from, and be able to like differentiate it from other things. We're going to talk about innervation to the gut, which comes both from the enteric nervous system, which is kind of just centered in the GI tract, and then also the central nervous system. So the central nervous system, like brain, spinal cord, can communicate with the gut, and the gut can communicate back, but the gut can also take care of itself with the enteric nervous system. It's really cool, the experiments they did to figure this out. And it's kind of like a heart, right? So we learned about pacemaker cells. If you just put them in a dish, they'll just start beating and beating and beating. I like to think of the gut being the same way. It's not necessarily beating per se, but it's able to function without the brain. Okay, last 2 things, be able to describe how proteins, carbs, and fats are digested and then absorbed. So we're not talking about absorption today, but we will talk about proteins and carbs and how they are digested, and fats are next time, just because that comes in more in the second part of the GI tract, which is just south of the stomach. And then last thing, this is all for next time, understand how absorption, secretion, and excretion occurs throughout the GI tract. So really today is just a great intro and get you started on some of the molecules you need to know. This picture I like to use as just the basic anatomy map of the GI tract, everything we're going to need to talk about, but it's also great to talk about fluid balance. So this is super intuitive, right? Like whatever goes in has to come out, and whatever goes in through our mouth or whatever secretions we put into the GI tract have to come out either through the feces, which is a very small portion of what actually is coming out of you, or it has to go into the blood, into the lymphatic system, and be absorbed for use by the body, okay? But here, let's go through the anatomy just so we know. So we have all these glands in the mouth, and these glands secrete amylase. So that's the first thing I'd write down, amylase, which starts to digest carbohydrates in the mouth. It doesn't do a full digestion, it just starts the process along, okay? It gives a little bit of a head start. So that's what your saliva is made of. And when, like, the Pavlov's dogs, they hear the bell, they start salivating, like they're getting ready to break down some food in their mouth. So that's like the first version of chemical digestion that we get. So once you chew something, it comes down your mouth through the esophagus, passes this point right here behind the liver called the cardiac sphincter, where now the food is called chyme, C-H-Y-M-E, chyme. And so now it's in the stomach, okay? And great, we know the stomach, that's the main place of digestion in the body. Stuff is getting broken down, both like the stomach is squeezing in a bunch of different ways, and there's a lot of chemicals in there. The pH is abysmally low, And then all of a sudden you go through the pyloric sphincter here and you're into the small intestine, all this mushy brain looking stuff in the middle. So you have the duodenum, jejunum, and ileum. Those are the three parts of the small intestine. You're not going to be like tested on this stuff specifically, right? This isn't anatomy, this is physiology, but it helps to know like where you are in the body, right? Then the last thing, the small intestine connects to the large intestine, this big thing just circling the outside. And then it goes through the rectum anus and excretes the feces. So that's the GI tract in general. Last two things you need to know here are the liver and the pancreas. So these are what are called accessory GI tract organs, and that food does not pass directly through them, but they do secrete specific molecules or enzymes that are vital for proper digestion. So we'll get into those more next time. That's not really the focus of today. So what we're really focusing on today is head, like the cephalic region, right? Cephalic is head. Cephalic innervation of the gut, what's happening when you see food, stuff like that. So we're talking about mouth down to the end of the stomach, and then next time we'll pick up with the small intestines, and that also includes the pancreas and the liver. Even though it looks like the liver would dump in the stomach, it dumps actually down lower into the small intestine. Okay, so stimulus to the GI tract is kind of what this slide is showing, and there's a lot of info here. I'm not going to go into depth with this up here. This is just showing you the layers of the gut. So the most internal one, so if I had like a bowl of, let's see if I can. Okay, so if I had food like right here along where all these letters are, the closest region would be the mucosa. Underneath that's the submucosa, muscularis, and serosa. I feel like you're tested more on that in anatomy than you are here. Just nice to know though, like what the layers are. Like if you were to cut like any part of the GI tract, you would be able to see these layers in a cross section, which is kind of cool. So let's get into the stimuli. So let's take this step by step. Don't get freaked out by the huge amount of graphs, right? So first things first, stimulus. So that can be pH, stretch, osmolarity, or products of digestion. So there's so many different stimuli, that can help us, not really help, but force the GI tract to get to going on its digestion, on its motility of the food. So there are two different arrows here. The red arrow stands for short reflex versus the long reflex, which is involving the CNS. So we talked briefly about the enteric nervous system, and there's a slide later on more about it, but the enteric nervous system is located in the GI tract, and it allows the gut to specifically manipulate itself if needs be. So for instance, if you stretch out the cardiac sphincter into the gut, all of a sudden the rest of the stomach can sense that. There's actually neurons that like travel in and out all around the gut. It's fairly highly innervated in that way. So they can be told you need to start digesting, they'll start squeezing, they'll start releasing all the chemicals they need. So the enteric nervous system is the little brain. So stimulus reaches here, you have sensory receptors, interneurons, efferent neurons, sorry. So this is exactly like what we learned in the reflex involving the spinal cord, except there is no spinal cord or CNS involvement necessarily. Now, if we do the long pass, so actually let's finish the short. So short enteric nervous system goes all the way through effectors, just being the smooth muscles, starting to get ready to move that food, or the chemicals are being released now. It's time to eat, it's time to digest. And then last thing, you start getting your digestive response. So if you do the long pathway, which both of these are happening, it's not like your body's picking like, okay, tell the brain, don't tell the brain, tell the brain, don't tell the brain. They're both happening. It's just the short reflexes can happen faster, obviously, right? And the long ones go up to the cephalic brain, which is your brain, right? So you're also having other stimuli that aren't directly involved in eating. So down here, this is all eating related, like the pH of your gut might change if you drink something really acidic, like orange juice, you stretch, the osmolarity goes up, all that's like stuff going into your GI tract, whereas the stimulus up here is like seeing the food, right? Like this would be Pavlov's dog. Like you see the food, you hear the food, you hear that bell, you hear the food, sorry, you smell the food, or you hear the bell. Now your sensory receptors are going and you get the same responses from two different systems. So I think that's pretty cool. Let's just move on here. So this is the enteric nervous system. We just talked about it. So there's intrinsic neurons and there's also extrinsic neurons, but the intrinsic neurons just mean that like there are nerves that specifically go from the GI tract to the GI tract, not necessarily going all the way from the brain down to the spinal cord, down to the GI tract. There are nerves like that, but that's a part of the cephalic response, part of the CNS response. So those two things are synonymous. So really the enteric nervous system, just know it exists. You're not really like, this is still area of research for a lot of people. It's not like we know everything that's going on here, but there are like specific neurotransmitters. So like serotonin. So for instance, if you take an SSRI, right, which is used for depression. That's a selective serotonin reuptake inhibitor that primarily acts in the brain, right? But some of it can actually get into your gut, and then you are secreting more serotonin. Serotonin is also a molecule that's involved in motility in the gut. So one side effect of SSRIs are diarrhea, because now stuff is too mobile and it's getting through too quickly. You're getting diarrhea. So just know that there are a lot of signals going on in here. We don't talk at all in this lecture about the microbiota, really. We briefly mention it with the large intestine, but that's huge too in understanding digestion. So enteric nervous system, just the nervous system that takes charge of the gut all by its lonesome. So this is a long versus short reflex. So if you think long, think, okay, that's food and brain and down. That's long. Whatever the food is telling the brain, and then the brain is telling the gut. Short reflex is literally the food gets into the stomach senses that, and now you have like your response from the gut. So if you want, take a minute, pause. You can go through like this chart here. That's totally fine. We don't really need to know too much about ghrelin, leptin, or the hypothalamic appetite center. That's fine. If you want to look more into that, I'm not going to really cover it because it's not super important. So yeah, take a moment if you want. Really, we already talked about what is shown here, like the long is through the brain and the short is not. Okay, so I'm going to move on. Okay, these two things I would know pretty well. So what is the difference between peristalsis and segmentation? So when you think of peristalsis, you want to think, I am moving food along the pathway in my gut. This happened all the way up from the esophagus down through the anus and rectum all the way until it's out. So this is super important, right? So I think I learned in one of my classes that they were worried, well, if we send people into space, will they actually be able to digest food? Will they be able to defecate? And they started doing research into it, and they found out, yes, they can, because food isn't just pulled through your system by gravity. Even if you're in a no gravity zone, your body can still take care of it. And it does that by contracting the smooth muscle layers behind the bolus of food, and then squeezing like a tube of toothpaste, moving that bolus forward. Okay, and it's not just like it's happening at one spot. It'll be happening here, it'll be happening here, and just little movements get that food all the way through you. Okay, so this really isn't involved in digestion per se, unless you consider like movement a part of digestion, which you can, but really I would consider segmentation the bigger part of digestion. So all that's really happening here, you're not getting this systematic movement. You're kind of just getting the smooth muscle contracting up and down and up and down and up and down. And what that's doing is pinching off different parts. So if you look at the screen is now over here, like after it happens, and it's with the yellow, and then it's also with the red, and the red is moved with the blue, and everything in the gut is being like mushed together, okay? So it's kind of like when you dump out your fridge and put it all into a pan and see what happens. Like that's what's going on with segmentation. You're just digesting all that food at once. And like it says, there's no forward movement. So know the difference in this slide, know the difference between peristalsis and segmentation, please. Okay, moving on. So this is kind of cool. We talked about this in the muscle lecture with smooth muscle. And it's the slow wave potential, meaning that skeletal muscles need direct innervation if they're going to fire. And we went over that in depth before. But smooth muscles actually don't. They can make their own potentials, kind of. And some of them will reach threshold, some of them won't. It's kind of cool how this works. So your gut is never just going to be completely still, right? Your gut is going to be moving. And if it's not getting like innervation from the cephalic or the enteric nervous system, then it's still going to be moving somewhat, like still making sure there's net movement through. You don't really want anything just sitting inside of you, like gum for 7 years. I swallow all my gum. So if that were true, I'd be dead by now. So I don't think that's a true statement, but just know that these smooth muscles are going to be functioning by themselves all the time. And it's not like this is doing something great for the gut. It's just preventing it from stalling out, from being still, okay? So this is, I think, the most important part of this lecture, maybe the second most important part, but we're going to talk about the cells of the gastric pit. So this is a pit, right? So what happens is this is the stomach lining. So gastric pit is only in the stomach. So you can picture like up here at the top of this slide, the esophagus dumps, now we're in the stomach, and this is the lining. So you have these big, big, big invaginations of cells that go down, and pretty much what we learn about invaginations in the digestive system is they're there to increase surface area and the activity of the GI tract. So all these cells down here produce different hormones or different active molecules that help with either digestion or protecting the stomach itself. So the first one we want to talk about is the goblet cells. Now you guys don't need to know like the goblet cells are on top and at the very bottom are the G cells, and the D cells are, you don't need to know the location of these cells, but you need to know specifically what they do, how they interact with one another, and all that kind of stuff. So goblet cells secrete mucus, and you're like, that's disgusting. I don't want mucus in my gut. Well, you do, because the pH of your gut is going to be around 2 when you're actively digesting, and that would destroy pretty much any living cell around it. So this mucus secretes a layer of like glycoproteins that can really absorb that hydrogen charge, and it's all the time renewing itself. Okay, so goblet cells, mucus. Okay, so I would know that. I'd be writing that down if I were you. Parietal cells, these are the main, in my mind, the main functional cell of the stomach, because they're what are producing acid. There's like a whole nother slide on that, so don't worry, we'll get more into that. But parietal cells, when they're signaled to fire, they release hydrochloric acid, so an H plus and a Cl minus, and that floats all the way up here, and that starts denaturing proteins so they're more linearized, and so that you can get peptidases to cleave them. And they can also have an impact in carbs, carb digestion, but mostly it's protein. So parietal cells release hydrochloric acid. Next are the chief cells, and they release pepsinogen. And remember, we learned anytime something ends with OGEN, it's the inactive form. Like we learned about that in the RAS system, like last lecture, I think, with angiotensinogen. So it has to be cleaved before it's active. So pepsinogen is released into the gut. And it's really important that it's in this inactive form, because if you were just releasing active pepsin, then you could actually be cutting up all these cells down here. So what happens is the pepsinogen floats up, not just floats, but is moved up, right, into the actual stomach. And then the very low acidic conditions, the low pH, is enough to cleave the pepsinogen. So parietal cells, HCl, and then pepsinogen turns into pepsin under HCl's guidance. Okay, and then last two, D-somatostatin. Just know that that's like, when you think of D cells in the gut, you think somatostatin and negative feedback. So these inhibit any more acid secretion, like we don't want it getting too low of a pH in our gut, and it will kind of slow us down. Super important. And then G cells, gastrin, which stimulates acid secretion. So that means this gastrin, so the G cells are at the bottom here, they move over, secrete gastrin. Gastrin talks to the parietal cells, tells them to release acid. That acid talks to the D cells, and the D cells try and turn that response off before you get eaten. So there's still plenty of acid produced that can start digesting things, but the D cells will stop that with their somatostatin. Okay. Now, so this slide is just a dumbed down version of this slide. And if I were you, I would know this slide pretty well, okay? So not everything, like we're not really talking about vitamin B12, but I would know substance secreted from each of these cells, okay? And then what their stimulus is. I would know most of the functions, like what is mucus used for, what is bicarb used for, all that kind of stuff. So if you have the time, I would recommend going over this chart. It's on Learning Suite. You can pause it right now and try and test yourself a little bit. But just know, like, somatostatin is secreted in response to too much acid, and it comes from the D cells, and it then in turn inhibits more acid. So if you have like a study group or study partner, I would just quiz each other on all these cells, specifically the G, the D, the chief, the parietal, and the mucus or the goblet cells, okay? Okay, that's all the time I'm going to spend on this, so you guys can go over this more, and please do. I want you to make sure you know this. It's just not really pertinent for me to just go over every single box here, though. So this is how the acid is actually produced in the parietal cells of the gut. So what happens here is you have the hydrogen potassium ATPase, and it looks like it's an antiporter, right? You guys should be able to say, differentiate between antiporters, symporters, and uniporters super easily now. We've gone over so many of them. So you have water that's broken down into H plus and OH, and hopefully you guys recognize what Ca is. That's carbonic anhydrase. That's formed bicarb. The bicarb gets pumped out in exchange for Cl. Then Cl comes over and both hydrogen is pumped out and Cl actually can just diffuse through its channel. It doesn't need active transport here. But what's kind of cool is that this channel is a CFTR channel. That's, you don't need to memorize this, I just think it's kind of cool. And that's the same channel that can be damaged in patients who have CF or cystic fibrosis. So we usually associate CF with like the lung problems and the heavy infections of the lungs, but they can actually have a lot of GI issues as well because the chloride can't actually come out, and that chloride helps with the acidity by bringing the hydrogen out through this ATPase. So I would know hydrogen comes out, you're using a antiporter for potassium, and chloride will also follow through, right? So chloride is following its concentration gradient and also the positive charge coming out of these cells. So that's how a parietal cell will secrete acid when it's stimulated to do so, usually by gastrin, right? Okay. This is the mucus barrier we talked about. And what's really cool is let me go back 2 slides really quick. I didn't really cover this, but these mucus cells, secreting cells, also make bicarb, which is super great because it's a buffer to the acid. So not only do you have a physical barrier, but I also like to think of like a chemical barrier, and like what's right around these cells is very basic, and what's up here is super acidic. So the mucus provides like that physical layer, and the bicarb provides a chemical balancing, like it neutralizes all that acid, right, by picking up that extra hydrogen and protecting these cells. So mucus cells can secrete both of those things to protect their own function and the function of the other gastric pits. Okay, this is, let's see, this is the last of the gastric pit things that we need to go over before we just really quick do protein and carb digestion. So there's a lot going on here. What you guys need to know is there's multiple inputs. Chief cells make pepsinogen, D cells detect like the pH, right, and secrete somatostatin. And how does that pH change? Well, it changes if parietal cells start to push out hydrogen, right? So negative feedback to the D cells produce somatostatin. Somatostatin shuts down everything, right? Shuts down it all. We didn't really talk about these cells. You don't need to know them. Dr. B might give you a question, but they're not super important for our purposes. And know that gastrin is released from G cells. So we've already gone over all of this really. You have inputs from the food coming in, the osmolarity changes, right? You have amino acids or peptides that can lower the pH, right? Because an amino acid is still an acid, right? Or you have input via the vagus nerve, which covers over 70% of the parasympathetic response in the body, and therefore stomach is under parasympathetic control most of the time. So I would take a quick minute, pause me for a second, go over this, make sure you get it, and bring any questions you have to lecture, but we have talked about all these steps so far. Okay, let's finish up with protein digestion. So you have a protein, right? And you have the N terminus here with the free amino, and the carboxy terminus here with the free carboxylic acid. And then we don't really care for the purposes of this class what all the in-between amino acids are. You do in other classes like biochem, you have to learn specifically like what amino acids chymotrypsin goes after, and it's like those ones with the big hydrophobic side groups. So we'll talk about that a little bit, but it's not the most important thing to know. So just know the difference between an endopeptidase and an exopeptidase. So an endopeptidase is cleaving these peptide bonds somewhere in the middle here. So trypsin, chymotrypsin, pepsin, they all have specific amino acids they're going after, and they're cutting in the middle. Okay, an exopeptidase cuts the very end peptide bond. So let me do it right here. So an exopeptidase would cut here. Specifically, this would be carboxypeptidase. And then over here, aminopeptidase would cut this bond. So it just finds the nearest end terminus, cuts it, and goes in from the edge. So these things work together. So think of it like this. So this could be trypsin cutting right in the middle here. So now you have 2 peptides. And now you could have an aminopeptidase working here, an aminopeptidase working here, a carboxypeptidase here and here. So now you're just getting the breakdown of proteins a lot faster. And you know these are huge molecules. So it's great that we have so much involved. And we're going to learn a little bit more about protein digestion next lecture when we talk about pancreatic secretions and what goes on in the intestine. But this will be a helpful slide to get us started off. Okay, let's do carb digestion. So like I said, you have amylase, right? Amylase starts in the mouth, and then it starts the digestion by just breaking some of these starchy bonds, right? And the starchy bonds are just a sugar to sugar to sugar to sugar bond. And you don't need to know like what maltose is, what sucrose is, what lactose is. This isn't biochem. What you do need to know is that there's specific enzymes for each of these sugars, but that's really easy, right? Like maltose, maltase, sucrose, sucrase, lactose, lactase. Like that's one thing biologists did for us that made our lives a lot easier, okay? So you ingest a lot of starch, you ingest like cellulose you actually can't break down. That's like from plants. It acts as a really good source of fiber in humans. Like cows can break it down, they can eat grass, but that's because of their microbiota. There's so many cool things going on in the gut with digestion. It's like an area of research for me, so I could just go on, but I'm going to stop so I don't bother any of you. But just know like carbohydrate digestion is amylase, and then it keeps going on in the gut too. This is a lot simpler than protein digestion because we have specific enzymes acting on specific sugars, and these sugars all have a very specific makeup. Like you have two glucoses bond together, a glucose and a fructose, and two glucoses. The difference between maltose and lactose would just be oh, I'm sorry, that's a galactose molecule in lactose. See, I've already forgot all my biochem. You don't need to know it for this class. And last thing I want to make you guys aware of is making glucose from smaller precursor molecules is known as gluconeogenesis. Highlight, bold, underline this term and understand what it means. So in times of fasting or when there isn't active supply and your blood glucose goes down a little bit, your liver will break down glycogen, which is the storage form of glucose, and produce glucose. But if there's no glycogen to be had, it will actually go through gluconeogenesis. It takes like amino acids. It can take other little molecules and reform glucose that it can then spit out to the rest of the body. So gluconeogenesis, making glucose from little molecules in your body. Okay, you guys, so that's it. We'll talk about the intestinal phase all the way through next time. I do think that might be a little bit harder, so please make sure you understand this. Email me with questions. Make sure to speak up during lecture if you do have questions so we can go over what you guys need, okay? So that's going to be it, and I will see you on Friday.